Thank you, Ellen. So welcome. Good morning, everyone. Today, my name is Thomas Hankemeyer from Leiden University. I'm co-sharing with Janneke.
Yeah, my name is Janneke Wijs from the Radboudumc.
And we will lead you to the first session. As Ellen mentioned, CosMx should be fun, but no fun without data and infrastructure. So that's what we will do in the first session. So we will start in the first session with the 4 pitches. The first will be Ies Nijman from Utrecht. Ies is co-leading the GeoMx pillar within CosMx. He was involved from the beginning, and then when Edwin left, he took over completely the leadership, you could say. So thank you for that. Ies has a background in DNA research, diversity, and so on, and moved then from the more animal field into the human field at Utrecht and Utrecht, and now leading not only the sequencing facility, but also the bioinformatics and the high-performance computer facility. So really facility manager. Ies, the floor is yours.
Thank you, Thomas, Janneke. Okay, I would like to introduce our sequencing facility, which we called as a part of the GeoMx facilities of CosMx. Let's see how this one works. Yeah, so as was introduced, so we are one of the pillars on CosMx, mainly the genomics part coexisting with the UMC Utrecht, my facility, and Janneke's facility in the Radboud. And basically what we offer is a broad range of genomics services, mainly focused on sequencing, DNA, RNA, and everything what goes around, so isolation, library preps in all matters. But also the sequencing has a large plethora of machines and equipment and approaches. And mainly we have the Illumina, obviously, as a sort of standard tool of sequencing. But also in Nijmegen, they have the PacBios running long stretches of sequencing, single molecule. In Utrecht, we have the Nanopore that nicely complements that sequencing strategies. The Radboud mainly focuses on diagnostics and high-throughput research projects, while in Utrecht we're focusing on the smaller projects, really innovative and ranging from low to mid to even high-throughput solutions. So after a while, and Utrecht is mainly working in a cancer setting in our department in the hospital. We did get questions, yeah, the sequencing is all very nice, we sequence in bulk, and now we can move a little bit into single-cell sequencing, but what we miss is really the context, and we want to look in the archives of the pathology, so FFPE material needs to be exposed to sequencing in a proper manner. So we really got the questions from the researchers, yeah, we need to bring the biology back into all this sequence data. And at that time, the really spatial omics field started to sort of develop, and we decided to jump into it also with CosMx and our facilities to acquire those equipment and then really offer this as a service as well. So you see these CosMx leaflets, they're probably in the front as well. Highlighting what we can do with this spatial technology. This is already outdated, so I updated the slide a little bit because the field is really moving forward. And basically we have two, oops, can I go back? I spoiled the nice pictures already. So basically we have two types of equipment. One is the Nanostring GeoMx, which is focusing on FFPE microscopy slides where you can stain your tissue. You can select regions of interest, really focusing on, well, this part of the biology I want to sort of investigate in details. And then you can go through the tissue and then either interrogate RNA panels, really up to the full transcriptome, but also protein panels. So you can really do a multi-omics experiment on focused sections of your tissue. And like the latest edition is a 580 panel protein where you can really detect on small populations of cells selected from your tissue. So we acquired this machine, I think, 2 years ago, and the first experiment which people did, the researcher came up, yeah, this is really nice, but I want to focus on that cell there, which I can see. So luckily the company realized that as well and moved also building the CosMx, which is now similar. It works on FFPE microscopy slides, looking at your tissue, but then really go into the single cell. So you can screen your whole microscopy area and then interrogate on single cell level RNA or protein panel or even a combination. this is not full transcriptome yet, so the biggest panel is now the 6,000-plex RNA, but the whole transcriptome is already demonstrated internally in Nanostring, so we will expect to have this in 2025, where you're able to look at hundreds of thousands of cells and interrogate a full transcriptome in combination with the protein panel. How does that look like? This is an internal collaboration with Hugo Snippert, where we look at early stages of colorectal cancer. So this is stained with a few morphology markers, and this contains about, from the top of my head, about 700,000 cells, where we interrogated with RNA panel, 1,000-plex. But actually we used single cell, or Hugo used single cell data, which he acquired from the same tissue to sort of create profiles of his typical cells, and then overlaid this data, and I hope this will show up, to really identify which types of cells you are now seeing. So not only on the RNA 1,000-plex you can identify the cell types, but you can bring in your own single cell data to really highlight those specific cell populations. Where you have single cell analysis, you see all your populations in your clustering, but now you can really map them back in your biology context. So I think that's really, really nice. So we had a little bit of challenge where we had some legal issues between Nanostring and 10x. So basically the last experiment I showed was also the first and then our last. So we were sort of paused for half a year, and actually luckily since a few weeks we could start again. So we did some experiments on protein level. So this is the same early colorectal cancer where we looked at a 64-plex protein panel, and zoom in over here where you can see a nice area. And then all these analyses you can do. So cell typing can be performed. I hope this will cross into the next as well. But then we have some more examples. So in the meantime, we said, well, how does it work on multiple tissues? So we decided to make a multi-tissue array of any type of tissue you show on the screen, you see on the screen. And we focus in on one particular example. So it looks almost like a planet, so we like these pictures a lot. This is tonsil tissue, where there are germinal centers which really produce immune cells, B cells. And on the right panel you see all the analysis you can do. So this is an example of neighborhood analysis. It jumped pretty quickly, but you can, I cannot even go back. But you can do a multiple analysis on this and pop up all the specific markers. So there was a picture in there showing CD19 for B cells, where you can clearly highlight these in the biological context of the whole tissue. So as I mentioned, we offer this as a service. So if you need help or are interested, we can help you set up or design an experiment, show you what we can do and what we cannot yet do, and really help to design your experiment. So Alain mentioned the email address, and you will arrive at our facility or at the facility of Nijmegen, and where we can sort of help you set up your experiment and do your experiment for you. That was what I wanted to say.
Thank you. It's very nice, and also, let's say, it shows the dynamic of the field. When we wrote the proposal, we couldn't dream of this kind of facilities and opportunities, instrumentations. There is time for one burning question, if you would have. Otherwise, Ies is, of course, around the whole day. If not, then before you disappear, is appreciation.
Okay.
Thank you.
So the next speaker is Kelly Stecker. Kelly Stecker studied biochemistry at the University of Washington and worked in the plant biotech industry for several years before obtaining her PhD in biochemistry at the University of Wisconsin-Madison. In 2016, she moved to the Netherlands for a postdoc in the biomolecular mass spectrometry and proteomics group at Utrecht University and began her work on protein regulation and protein expression in cancer, with a specific focus on signal transduction events that regulate tumor immune cell interactions. She became an assistant professor in the BMSP group in 2021. Kelly's research focuses on applying innovative MS approaches to understand cell signaling and molecular interactions in immuno-oncology. And she will now tell you something about her facility.
Yes, thank you. Thank you very much. So I'm an assistant professor in the biomolecular mass spectrometry group, and there my role is to act as the facility manager for our CosMx initiatives. Let's see how I click. Yeah. And together with the Radboudumc and the NKI, we offer proteomics facilities to the CosMx community, and our goal is to enable your research by sharing our expertise and our equipment with you. And some of the services that we offer as a proteomics facility are outlined here. It's a bit small, but you can also see it on the website. We can quantify the proteome of complex and very low abundant samples. If you're interested in cell signaling, we can quantify the phosphoproteome. And we can also do affinity purification analysis if you're interested in looking at protein-protein interactions from your samples. And to do this work, we apply various quantitative methods, and we have lots of high-end instrumentation, including 3 Q-Exactive Explorers 480 mass spectrometers, and a Fusion Lumos tribrid, and a timsTOF instrument. So that's what we have at our location, but actually this slide is already outdated because this year we were able to make some new investments into mass spec instrumentation, which allow us to address some of the current challenges in the field. So at the NKI, they have purchased an Orbitrap Astrel mass spectrometer, and this is the newest, one of the fastest, most sensitive mass spectrometers, which is really streamlined for proteomics applications. So here they're working on single-cell proteomic workflows. And at Utrecht, we've invested in a new tribrid instrument called an Eclipse, and this allows us to do novel protein sequencing workflows, which are really important for understanding the sequence of proteins, including intact proteins, which is relevant for my colleague Danique's presentation later in the day. And this instrument also lets us do intelligent data acquisition. It lets us be smarter with how we want to acquire our data on the instrument. So to give an example of our proteomic capabilities, our quantitative methods, and some of these new techniques, I thought I would share a little story with you today from some work that we're doing in our group. And together with the NKI, we became really interested in the communication dynamics between cytotoxic immune cells and their tumor targets. And of course, the reason that we're interested in this interaction is it's really the foundation behind immune checkpoint inhibitors that act to reinvigorate the immune system to kill cancer cells. So ICB therapy has been revolutionary in cancer therapy, as you probably know, but resistance is a big problem. So the idea is that if we can study the communication between a cytotoxic T cell and its tumor target, we can start to understand mechanisms of tumor resistance. So our collaborators at the NKI have looked at RNA expression to understand this system, but of course, if you want to study signaling, proteomics and phosphoproteomics is a great approach. So here I'm showing a video of a cytotoxic T cell in red engaged with a tumor in blue, and what you can appreciate is that this is a real physical interaction, and these cells are difficult to separate for analysis because they're attached. So we have a challenge here. We want to study the signaling dynamics occurring between and within 2 different cell types, but of course when we lyse our cells, we lose the cellular context. So how do we do this? So a strategy that we adopted is that we use isotopic labeling to uniquely label the proteins from each of our cell types. So tumor cells are labeled to be heavier than the immune cells, and when we culture these together, we introduce a 3rd label. So that means that when we look at these proteins on the mass spectrometer, we get 3 unique pieces of information for every single protein. We can answer the question, is this protein expressed in a T cell, in a tuber, or newly translated? And that's also true for the phosphorylation sites that we measure. So we barcode our cells this way, and then we turn to a second quantitative strategy, in this case label-free quantitation, to follow these barcodes across our different samples. And this analysis allows us to quantify protein and phosphorylation dynamics occurring in T cells, in tumor cells, and in new translation. And this works for short-term interaction studies, and we call this method HiSeq for hybrid quantitation of cell-like interacting cells. And to give you a flavor of what this analysis can tell you, so we took a primary T cells and co-cultured them with tumors, and we looked at 4 different lung cancer cell lines, and over a short time course measuring 2, 4, and 6 hours of interaction. And if I look at all the phosphorylation responses that are specifically happening in tumor cells when they're attacked by a T cell, I can cluster that data. And it's a lot to interpret, but the point is that if you look into these different clusters, you can see what's going on in the tumor cells. How are the tumor cells trying to defend themselves against the T cell attack? And here we see different pathways, and what I want to highlight today is that, for example, we see that ROD-RAC GTPase signaling is activated, and in the data we see a particular effector protein in this pathway, PAC1, is being activated. So if we go back to our co-culture, and now we co-culture the T cells and the tumor cells in the presence of an inhibitor of this protein, a PAC1 inhibitor, and we look at how the tumors now respond to T cell attack, what we can see in blue is the tumor cell growth on the y-axis over time under control conditions. In the red and the green are the individual treatments of the inhibitor or the T cell, and then in purple is the combination treatment. And you can see in the lung cancer cell lines we looked at, or in different cell line models for colon cancer, for example, we can see that we can further sensitize tumors to T cell attack. And we got to this functional target by interrogating the phosphoproteomic data that we found occurring during signaling. So that was very exciting, but we thought that we could do better, because actually, due to the different size of these cells and their different proteome content, we significantly undersampled our T cells in our co-culture experiment. So here you can see a number of phosphocytes and proteins were measured for each cell type. We wanted to get more information on our T cells. So to do this, we turned back to our co-culture system, and now we label our cells, like I said before, and now at each co-culture, each time point in each cell type, we apply a stable isotope tag, an isotope tag, which allows us to multiplex our sample, okay? So we can highly multiplex our co-cultures all together in one. And the reason this is beneficial is now we can use the new instrumentation to interrogate our data real time. So we identify the proteins, the peptides that are coming from all the content in our mass spectra, and then real time during acquisition, we sequence these peptides, and we ask the question, is this peptide a T cell peptide? If yes, then quantify. So here I am using real-time database searching to select specifically a cell type that I care about in my mixed population. And this really worked well. You can see that how much more content we got from each of our target cell types when we use this strategy. So, and for example, just increasing the amount of T cell proteins we can quantify, we can now look at these particular pathways I mentioned that before we couldn't see in our undersampling. So to recap, we are showing how to use quantitation in proteomics in a different way that allows us to look at 2 different cell types in a co-culture model. And this let us identify functional targets to sensitize tumors to T cell killing. And the application of real-time search lets you improve sample coverage for whatever your target population is. And there's really a lot that can be done with these intelligent data acquisition strategies, and I know that Alain's group at the Radboud is investigating this, bringing it to new instrumentation platforms. So I think it's an exciting field. And with that, I want to thank the biomolecular mass spectrometry group, which is a great place to work, the postdoc that worked on this work, Alexa Carey, and our collaborators at the NKI, and we are open to discuss with you any proteomic strategies that you would find interesting through the help desk. So thank you very much.
Thank you very much for this, explaining this nice technology. There's maybe time for one urgent question. And if not, then she will be here all day, I guess. So you can meet her. Oh, sorry.
Good, then the chocolate, yeah.
The chocolates.
So then I want to introduce the next speaker, Amy Hams. She is also coming from the US, did there her PhD postdoc with Dick Smith, and then went also to Wisconsin University to lead there the mass spec core facility, doing mainly proteomics, but also metabolomics and other things. And then moved in 2010 to Leiden to lead there the Metabolomics core facility at that time, part of the Netherlands Metabolomics Center, and then continued and now co-leading the metabolomics pillar within CosMx. Amy, the floor is yours. We need the other presentation. Yeah.
Okay. Thank you. So my goal now is to present what the Metabolomics pillar in the CosMx project is up to. The Metabolomics facilities in the CosMx project combine the expertise from several facilities, including Leiden and Radboud and LUMC. I have slides for those to present their work. The metabolomic services offered by the Metabolomics CosMx include a lot of advice for how to set up your project, how to do proper sampling, how to do proper storage, and we will advise you on how to set up a good experiment that will be appropriately powered so you'll be able to use your data to come up with the answers you'd like. We also then will do measurements and we'll give you a report and follow up with help with data analysis and statistical analysis and help you actually use your data to solve your biological problem. The Radboud Metabolomics facility has several technologies that include targeted and untargeted. They have computational tools that will allow you to get the answers out of the data, and they specialize in patient materials and organ on a chip in order to find biomarkers for rare metabolic diseases. At the LUMC, Martin Kira and his group do translational metabolomics and especially lipidomics. They have targeted, untargeted, GC for polar, volatile, apolar, a lot of lipidomics methods, including NMR and imaging. So they're really experts in lipidomics. They work with cell material and with clinical material and have some very impressive output. My facility is the Leiden Metabolomics and Analytics Center, and we support metabolomic studies for clinical and exposome applications. The kinds of platforms that we offer include targeted and untargeted. We have a global reverse phase that has 2 injections for the polar and the apolar metabolites. We have a GC Orbitrap for the volatiles. And then we have some targeted platforms, including signaling lipids, which you can see from, this is the signaling lipid platform with several innate and adaptive immune response and more. So these are generally low-level compounds that we use to target these biologies. And we have other targeted and tracer-based metabolomics platforms, including biogenic amines, acyl-CoAs, and some tracer-based where the methods have been modified to include the isotopic labeled compounds. We've recently had the opening of our exposome scan facility. And in this exposome scan, we hope to have a better profiling of the exposure markers. In this work cell, we have liquid handling, we have centrifuge, we have a 4-degree incubator so we can store our samples while we're doing sample prep. And this little guy, you saw the movie, he picks things up from the work cell and puts them in the autosampler and starts the mass spec. And then when they're done, he picks the plate out of the autosampler and puts it back in the work cell. So this allows us to take on larger studies and have more automation, less boring work for the technicians. This work cell also has the advantage that it has a controlled environment which protects the samples from lab contaminants. What we don't want to have is exposure to the lab. We want to have exposure of the people to their workplace or something like that. This also protects the technicians from the hazards associated with the samples we receive. So we're very excited about applying this automated sample prep to our platforms. The exposome scan project, together with Utrecht, we want to measure a lot of exposures from semi-volatile to apolar. In the GC Orbitrap, we've been working with several other GC Orbitrap groups in order to get better libraries. The current GC libraries are often built on quadrupole data and they're low resolution. The fragmentation in an Orbitrap doesn't really match them, so we have to develop a new library for searching the Orbitrap GC data. For the global platform, we have been working with a triple TOF and with a xenotOF. We use swath in order to get data independent analysis done, and we have EAD for advanced fragmentation. So both of these are designed to give us data that will help with identification of unknowns. When you have an automated data acquisition, you're going to need automated data processing. And in our lab, we've been developing a real-time QC monitoring tool that will flag if the instrument is running out of specs, and so that we can stop the run and have a technician pay attention and not waste samples to it, just keep on injecting. So real time is important when you're doing too many samples for a technician to monitor. We also have automated peak alignment and peak integration. When the studies get larger, you're going to have to do maintenance on the instrument in the middle of your project or change columns in the middle of your project. So you're going to have some way of aligning the different batches over time. And finally, we run all of our data through MZ quality to make sure that the quality is preserved and our reports are going to give you the best possible data. So Anne Charlotte in Utrecht has been building a computational data processing workflow for identification. And she's been doing that using the in-house developed libraries from standards that we have and combining that with public MSMS databases. She has a tool that integrates with our current data pipeline that will match unknowns with libraries for identification. And moving towards the future, there's a research line with Ahmed together with Sylvia where they're integrating imaging and single-cell multi-omics to be able to do more single-cell work. In this work, they can sample out of 2D cultures or 3D cultures, including the mimetas organoplate graft. From these, they can sample and electrospray individual cells to find out their central metabolism and lipid composition. Or they can follow drugs to their metabolites, and they can find out which cells are taking up drugs and which are metabolizing them. So to summarize, the Metabolomics and Exposomics in the Metabolomics and Analytics Center in Leiden, their goals are to measure the actual health state of people and cell models, to understand the immune system, the gut microbiome, central metabolism, biomarkers, biomarkers of food, biomarkers of drugs, biomarkers of environmental hazards, and more. And some of the projects that we're working on right now include grip on MASH. And that's with Manuel Castro Cabezas, and that's for prevention and care of metabolic dysfunction-associated hepatitis. And Hypermarker, which is personalized pharmacometabolomics optimization for treatment of hypertension with Rick Robet. We're generating the Dutch exposome reference cohort together with Roel and Sasha to unravel the impact of non-genetic factors on our health. And with Elena, we're working on solutions that contribute to healthy aging and increased vitality. And if your project should be on this list, you should contact Crossy, and Crossy will put you in contact with the metabolomics specialists or combination of omics specialists to help you get set up to use our facilities. Thank you.
Thank you, Amy. Is there any urgent questions? Otherwise, we refer to the break. And the chocolate, Amy.
Again. So next speaker is Sander Bervoets, and he studied bioinformatics at the VU University. And after that, he came to the conclusion that there is a lot of algorithms available, but implementation and using those is more complicated, I would say. So his mission now is to help biomedical researchers in supporting and getting the best algorithms and analysis. And he's coordinating the Radboud Technology Center of Bioinformatics, and I guess he will tell you more about this.
Yes, so my name is Sander, and I'm the facility manager of the data analysis integration and data stewardship for the CosMx project. So yeah, we have a very wide field of many experts. So because the data is generated in many different core facilities, there are a lot of experts also there. And combined, the strength of the network of CosMx is leveraging all these experts. And it's my honor to be able to get all these people together and work on the data. We have several different services and support options. So we can help with crunching the data, getting the data from the instrument, processing the data to a little bit more processed variant. We also do downstream analysis, so there's more the statistics, the interpretation of the data. But also we do multi-omics data stewardship and integration, so that we have basically all the elements of your data, all the sample information and metadata all in one go, so everybody can work on it and it's well managed and also ready for publication later. So you can check our website here. Also, we have a QR code. And my pitch is a little bit different because multi-omics analysis is complicated, and my time here is short. So I would like to give you a couple of elements of success that make a data analysis project work in a multi-omics environment. And these are principles that I would like to convey to you, but also in principles that we do at the data facility. So first of all, keep your hypothesis simple and harmonious. So it is tempting to look at pathology, if you have a complex pathology, to test as many things as possible or get as many samples of your patient group in the dataset as possible. But we notice that if you keep it simple, you will be able to find those small signals better in your data set. So that's something I would like to mention beforehand. Also, it is good to have a balanced setup of your experiment, a balanced set of your samples as well. Like for example, one of the batches that we'll always find in multi-omics analysis is male-female samples, for example, in patient data, for example. And if you have a big range of qualities in you have samples that are better than others, it's good to have a lot of redundancy built in. Like it's expensive to sequence or process all these patients, but As soon as you lose part of your sample because the data is less good or the sample is less good, then you also lose the balance in your sample. So please think about that in the beginning. And this is obvious, but to minimize batch effect, please sequence the same sample, process the same sample for multiple experiments, and keep them in the same batch. We can correct for batch effect, as you can see here. We do that at the facility, but you will always lose some signal. So I just want to impart that extra emphasis for you. Now, something I always recommend is start fair. Start fair at the source. So make your data findable, accessible, interoperable, and reusable right from the start. As soon as you have the data, put it on a repository. As soon as you have samples, put them in the metadata scheme so that's available. Also for later in the studies. So it's also easier to collaborate with other people from other facilities and other universities to do your analysis better. This is important from the data side. So the more raw, the better. Of course, it's good if you have a very good pipeline to do some interpretation, but in the course of a study, like new tools will come out, like machine learning is a big thing, artificial intelligence is included now. So as long as you have access to the raw data, that's best. Sometimes you cannot avoid it, right? Sometimes you have to do some interpretation to just get the data files out of the instrument. Well, then you get a little bit of what I would say medium rare. Keep it medium rare. Keep it as, as plain as possible. Another thing I would like to focus on, this is something also we help with at the data facility, is open and linked data formats. Is that, well, you can put your data in a PDF, but all of you know that will be a problem later. An Excel file is better, but it's even better if you have files that have built-in linkage with your samples, with your metadata, but also between the data sets, between the genes. We can also help with that. Like, for example, the Fair Data Cube is one of the tools that we developed to make multi-omics data integration better. Something that we also really like to do is use open source tools and workflows. That's something we really focus on. If you want to collaborate over different facilities, different universities, if you have software that's behind a license, it becomes a bit difficult. And if you focus on open source tools and workflows, you can also do a lot of very good versioning. So if new tools come out, it doesn't screw up your analysis. That's something that we do and something that we also will happily do for you at the facility. And then lastly, data stewardship is when it all comes together. If you do active data stewardship, you combine all the elements I mentioned before into a comprehensive flow. And it's easy to onboard new people or new researchers, easy to publish as well. There are several more technical parts that we have, for example, the metadata registry and the Fair Data Cube and other infrastructures like a collaborative digital work environment for research. Those are technical aspects that not every university can have. Something you can always advise to have it, but if you do active data stewardship in a project, it's easier to get to this happy flow. So we have demonstrated the capabilities to do this. If you do everything well, you can get to very significant results. This is a workflow that also includes the analysis for downstream analysis, for example, as a pathway analysis. You can go to the GitHub and see what kind of steps were involved with this project. And just to emphasize everything, these are the points, and we will definitely apply these principles if we can help you with your project. And there are some links down in the bottom for papers that we published that also discuss these points. And of course, my question is, how can I help you? How can we apply these principles for your project?
Thank you, Sander. So there is a question.
My name is Marianne Beekman from the UMC in Leiden. I wondered whether you also are in contact with the data providers and how they can provide fair data at the source, because as a data receiver, I usually get no ontologies with my platforms, and as a researcher, I don't know anything how to make ontologies out of these. So could you comment on that?
Yeah, that's true. for core facilities, it's relatively new to be able to do these things. The way I see it is, if you would do a CosMx project with us, with the CosMx initiative, I will make sure that also we start linking this data right from the source, and that means also coordinating with the core facilities within our network to do that. So it's like I said, it's hands-on, but I think it's definitely possible if a data stewardship facility is immediately involved with the project. So that would be what my suggestion would be.
Hi, Erik Reitz, Amsterdam, UMC. Monday morning, very early, so a tricky question. Any words for advice for new multi-sited infrastructures, how to handle this? You're now on the way, but would it be something that would you ideally at the start would have done different to align these different data storages, combinations better?
Yeah, so that's a very good question, and it's actually quite tricky because if you do multi-site, you also are dependent on the IT infrastructure and the principles that are important for the separate cores and separate universities. I think my main takeaway would be to create a collaborative digital working environment right from the start. If you get that right from the start, that when you also make sure that all the legal requirements are there and it's safe and everybody can log in, then you start off with one place where the data is for all facilities. And that can be tricky, of course, like do you do it in the cloud? Are you going to get like an external provider to do it for you? Those are tricky questions, but if you focus on this first, I think the rest will follow more automatically.